It's wonderful to be here. Really great to interact with many of you, and it's beautiful to see the campus. So wonderful, it's a wonderful initiative. So thanks to the organizers. So I wanted to tell you about, so I think I'm the only neuroscience speaker at the end of the day, because I think Gautam's not talking about neuroscience. So I'll compress some neuroscience for you. I'm not going to be talking about too much about a lot of neural data, but I will be talking about this intersection of interests of mine, which is neural coding and neural dynamics, but also the emergence of structure in brains and biology. Okay, so specifically, I'm interested in this question of how brains develop very structured circuits, and interested in the mechanistic questions of how those structures emerge over development. Also in the questions on the evolutionary timescale that are more related to function optimization. So what are the functions that are optimized that gave us these circuits in the brain? And also then going beyond brains, which is if you just think about what are general principles for modularization, that's another interest. These questions apply equally to deep learning as well. And so I just won't have a chance probably to get to there, but if I do, I'll try to cover as much ground as possible. But the most important thing is that you can ask me questions while we're discussing, so that if I cover less stuff, that's fine. I'd rather that you sort of absorb anything you want to absorb in greater detail, so don't hesitate to interrupt. Okay, so this is work that's done by a bunch of really, really, really strong students and postdocs. Sidhartha is a postdoc of mine who is in fact probably this week or next week coming to start his own group here in ICTS. Mikhail was a PhD student. Rylan was a master's student, now a PhD student in Stanford. And Trevor was a PhD student who now has his own company, Extropic. All right, so the first thing that I want to just sort of philosophically discuss and point out is this idea that we have a lot of modular structures in biology, and modularity, I would argue, I would state really, is the essence of biological function. Okay, and modularity is what allows us to be efficient learners and efficient at evolving as biological organisms. Okay, so what does modularity do for you? Well, modularity has been posited to do a lot of things. So one is that modularity is posited to be something that gives robustness to perturbations. So for example, if I have a system that's computing many different functions, and if there's noise or perturbation to one part of the circuit of the system, it's not going to affect the functioning of the other systems. And so if the system were arranged in a modular way. It also can allow for the efficient evolution of complex systems by allowing independent modification of individual modules. So for example, if I had independent control over my finger length, and maybe something else like my skin color, then it would be possible to actually evolve those two traits independently, and presumably that would be an advantage to have somewhat decoupled and independent or modular control of those unrelated traits. Okay, then the other advantage of modularity is that if you do have a factorization or a specialization of function into different components or modules of a system, then it's possible to do things that involve compositional generalization, right? You can, if you have subcircuits that do a bunch of subfunctions, now you can have a system that can combine those subcircuits in different ways compositionally to do a whole bunch of new functions, right? So it's kind of like what's called like using finite means to generate infinite variation, if you will, right? So that's another advantage of modular organization. And that also means that it's possible on the flip side that you can do some, you can do learning in a sample efficient way, because if I've got a decoupling of functions and controllers, then it's possible to actually learn from data, right? Like if there's one function that depends on some subset of variables, you could learn the parameters of that function without having to set all the parameters of all the other functions. So that's another advantage, right? And so basically this whole idea is that you can tinker with components without redesigning the entire system. And then finally, from the perspective of scientists, it's kind of nice that systems are modular because that makes them interpretable. If everything was a big bowl of spaghetti, inputs in, outputs out, without modularity, it would be much harder to interpret, right? And Rama talked about like machines where you've got like separate components that do separate things, right? All of that is closely related. It's the exact same thing, right? Machines are modular. That means that you can like tinker with different parts of them. He talked about sectors. You have areas that recognize a substrate. They might stabilize the protein, and so on. Okay, so there's a nice parable here, which I'm not going to take the time to read, but you presumably have read it while I've been talking. And this was just, a point made earlier by Herbert Simon on how modularity can lead to, I think it's basically a parable on evolvability. Okay, and so, there's a bunch of, more theoretical works that I won't have a chance to talk about. Here we talk about how theoretically modularity can lead to this, so it's basically supporting this claim that if you have modular organization, it can lead to sample efficiency that can break the curse of dimensionality in learning. So that's one problem over here. Another type of work that we've done is try to understand how modular-like codes that we see in biology can actually be used for things like error-correcting codes in quantum computing. So this is a collaboration that I had with some people, Ike Chuang and others who work in quantum computing. Okay, so all right, so now let's ask a mechanistic question, right? So there's many questions that arise, if you're interested in modularity, right? So one of the questions is this mechanistic question of, let's assume evolution has determined that it's important to organize systems, biological systems, in a modular way, right? How does it encode that information and then decode that information to create a modular system? Okay, and so that's the question. Okay, and so there are so we can consider how for example, so I'm interested in the brain, and so I want to know the question. I want to know the answer to the question: how does a modular structure that I see in the adult brain of some animal, how does it acquire that modular structure, right? And so here's a perspective from it's now very common to build models of the brain by training neural networks, right? So you say, well, the brain is solving some problem. I'm going to build a model of how the brain solves this problem by training a neural network with some inputs, and some outputs to solve the same task, and then I'm going to ask how it represents things, and whatever I see as the representations in that circuit may be how the biological system evolved its structure. Okay, right? So, and then there's this debate, so within that paradigm, of course, people in computer science will argue about whether the kind of learning data that the brain uses is, or their, our artificial neural networks use is reinforcement learning, supervised learning, unsupervised learning. So this is a meme from Jan LeCun. And of course, the idea being that RL is the cherry on the cake. The joke is on him because now RL is used on lots of things, which he agrees with now, finally. Okay, but on the other hand, so these are, okay, so then on the other hand, the perspective of somebody who does molecular biology, right, like a geneticist, they might say, well, look, genes are the things that are encoding the modular structure that eventually is then decoded like a blueprint. It's basically the genes are lists. It's a list of ingredients. It also tells you how to assemble them. All of that happens. So it's really this bottom-up genetic coding that then basically sets up all the structures that you see in an adult. Okay, so so that's the computer scientist, that's the geneticist. But there's something really missing, I think, in the middle, which I think as physicists we'll all agree on. So before we fill in what's missing, let's just ask this question of like, what is the nature of genetic information, right? So basically over evolution, you get the environment, genes give rise to some phenotype, and then there's like some selection, and then that in turn comes back and shapes the genome, right? And then the question is, is the genome like basically specifying, like basically a blueprint, like a very high information code? Is it telling me about the shape and position of every finger, in my, hand bud, the connectivity of all pairs of neurons and so on? But of course, this is silly because then it's like, we've just moved the problem back of like, how did all the structure form in the brain? Then we say, well, it's all encoded in the genome, and then there's like really elaborate structure in the genome. So The alternative view is that, maybe the genome doesn't really act like a blueprint, really it kind of just sort of sets some permissive conditions and maybe, sets up some, it allows growth to happen. So there's a lot of other stuff that happens, but the genome is setting some very basic preconditions, like some initial gene expression patterns, maybe some parameters, maybe some gradients. Okay, so these are two extreme views, yeah?
Like there's a third picture as well, right? Which is that, okay, there's this first one where it's just you stored everything.
Yeah.
There's a second one where you're saying you could store initial conditions or parameters. But it feels like maybe there's a third one in which the genome is in some sense storing like some sort of algorithm. The initial conditions maybe just come from the cellular environment in which the genome is embedded. Is that
right?
Orthogonal, or is that
no? Absolutely, and you're totally right. Like there's a whole continuum here in the middle, right? So indeed, there are growth rules. So yeah, it's specifying by specifying the parameters of the growth rule. Presumably, it's kind of like setting. It's like specifying the growth rule. If you think of it that way, and right, absolutely right. There's clearly some continuum. In fact, that's exactly. I think the point that you're getting to is there's sort of like this there's this gradient of how much information could be contained in a genome, and what is the level of specification that the genome provides for structure and its formation. And of course, that's kind of like, there's a lot of sort of intermediate stuff too, right? Like, so there's sort of how much information does the genome provide? Is it big or small? But then if it provides a small amount of information, then a lot of the structure that forms has to come from processes that are unfolding right from the genome. And I think a developmental biologist, obviously, I'm just giving you the most naive cartoon of what people in different fields might say, but obviously developmental biologists have studied the whole time. That's their whole point, is to study how rules can unfold through interactions with physics and chemistry and so on, and give rise to structure. So basically, here we're arguing about the cake. The geneticist says, so the question is, where does which parts of the cake, icing, cherry, etc. learning is. The geneticist says, well, hey, where did the cake come from? Genes of the cake mix. And then the point is that if you've ever eaten cake mix, it's a very far cry from the cake. And so really, it's the process of turning the cake mix into the cake, which is the this dynamics and time and all of this, the processes, the dynamics of the system, right? And so this is the notion of emergence that we understand very well as physicists. Okay, so basically then the argument is that really the genome has relatively compact types of information in it, and then all of the organization happens through this emergence. Okay, so all right, so that's all very philosophical and well and good, and so we can think about just skip the water analogy. Okay, so let's go to a concrete system, right? So that's enough philosophy. Let's talk about this kind of cell, which is an amazing neuron that's found in the brain, and these cells are called grid cells. And so what you're looking at here is, this is a view of an animal running around in an open field. Okay, there's no local landmarks, but there's some global orienting cues. Okay, so the animal just runs around. The black is its trajectory. And here, actually, I'm showing you real data, right? So this is the trajectory, real trajectory of the animal running in the box. And all the red dots are the spikes of one single neuron, okay, as a function of the position of the animal. And what you can see is that these dots actually fall in a regular pattern. They fall amazingly on the vertices of a triangular lattice that's tiling this plane. Okay, and so these cells are found in layer 2-3 of the entorhinal cortex. So these are cortical neurons. Entorhinal cortex is a pretty high-level cortex. It's sort of all the sensory hierarchies, go through cortical processing, and then they end up in high-level cortex, association cortex, which is what entorhinal cortex is, and then association cortex then pipes into hippocampus, which is the center for learning and memory in the brain. Okay, so it's a pretty high-level brain region, and it's pretty amazing. I just want you to take a moment and absorb how incredible this is. So first of all, it's sort of like it's a crystalline pattern that exists in the brain, right? Messy components, beautiful crystalline geometry. Second point is that there is no sensory stimulus that is present at those vertices, specifically driving this response, right? So it's internally generated. Third point is the animal may be running, what does it take to generate this response? Well, sometimes the animal's down here running north, and sometimes the animal's up there running south. But every time it runs through this position, it has to know it's at that position, and that cell has to know so that it can emit a spike at that point. Okay, and the visual input is completely different when it's running from south to north or north to south, right? Completely different. Its speed may be completely different. It may be taking a fast or a slow trajectory. It may even be stopped and like eating something while it's doing that, right? So it's something which involves internal structure emergence, right? Some pattern formation that gives rise to periodic patterns. Moreover, it requires instantaneous updating and estimation of position as the animal's actively running around in this featureless environment. Okay, so it means, and the only cues available to the animal when it's running around in this environment about space are its own self-estimates of movement, right? So it knows how fast it's moving, or it can at least estimate how fast it's moving by, its physical, movement, moving its arms. It's got semicircular canals, so it has an estimate of velocity, which it has to then convert into an estimate of position so that that cell knows how to fire, right, and when. Yeah, go ahead. Fantastic question. So it turns out that if you look at, you place an animal into a brand new environment for the very first time ever, the cells will already be firing in this periodic pattern from the very first moment. But it's true that the pattern is a little blurrier and then it slowly refines, right? And yeah, so we can talk more about that. Okay, some points to make here is that, yeah, okay, so that's, and these cells have been found not only in mice, which is where this, in rats, which is where this experiment was originally done, but they've subsequently been observed in mice, rats, bats, monkeys, and humans. And in fact, the discovery was awarded the Nobel Prize pretty recently in 2014. Yeah?
Is the pattern always hexagonal, or it can be different depending on the shape or the environment of the box that the rat has been?
Yeah, excellent question. So when an environment becomes sufficiently familiar, then in both circular and square environments, they find the same geometry of the lattice. So it is still an equilateral triangular lattice. And it's also true that if the animal is familiar with an environment, then if the environment's scale, like size is different, like different environments are different sizes, the periodicity of the lattice is somehow internally determined, not determined by the extrinsic size of the environment. So in a larger environment, it'll just tile that larger environment by just placing more periods rather than changing the period.
And this kind of properties, is it across all kind of animals, or it's just on rats?
It's all of these animals.
The lattice shape.
That's right. Yeah. Yeah, that's also a very good question. So when I say that the response is a triangular lattice, it's when the bats and monkeys are, well, in the bats' case, when their actually wings are, tied so that they can only crawl in the 2D space. It's harder to do the experiments while the bats are flying, but they've been done. There's still ambiguity on how to interpret and understand those experiments. The cells that they record from this region while the bats are flying don't seem to be, they seem to have multiple fields, like here, okay? But they don't seem completely periodic in 3D. But we also don't know if these are grid cells. So it turns out that these 2D grid cells are only like 10 to 20% of all the cells in that area. And so it's possible that they were recording, they didn't first establish that these were grid cells in 2D and then see how they're doing in 3D. So it remains to be determined, but it's a really good question. Yeah.
I haven't quite understood this picture. Can you say what does a red dot mean and what does the line connecting dots mean?
The black line is the trajectory of the animal as it's running around. And so that's all. I'm just showing you the animal's trajectory. With top-down view of the environment, right? So this is the environment, and we're just looking from above. So this is a square environment. We're just looking at it from above. Black is the actual recorded trajectory, XY coordinates of the animal as it moves. And all the red dots come from a single neuron and these are the spikes emitted by that neuron along the trajectory.
So then what does the lattice mean? If it's a single neuron, then what is the lattice?
The single neuron is firing at all of these locations. It's firing a lot in this neighborhood, it's firing a lot in this neighborhood. So, and then if you look at the collection of the neighborhoods that the neuron is firing in, those form a triangular lattice. Yeah.
In these vertex points, so we see more red dots. So is it some sort, can we interpret it as some sort of decision that they are making?
Yeah, I mean, they exactly. I mean, they've decided, they know that they are at this position and not this intermediate position, right? That cell knows because it's somehow estimated its position and has decided it's going to fire there, right? And then it's going to fire there, and it has to know that it's not there, right? When it's there. So it's pretty amazing. This is truly cognitive code, right? This is not a sensory-driven neural response because there is no external stimulus that can tell these neurons where they should be firing to form that pattern. Okay, so it is an internally generated response. Okay, so truly a cognitive code. Yeah.
Why that one neuron? Is there one neuron special? What, like, because, from what I understand, there's a lot of neurons in the brain, and so for behavior to happen, I'm guessing a lot of neurons would have to fire at once.
Yeah, so this is oh, yeah, that's a good question. So this is one neuron from this layer 2-3 in entorhinal cortex. If you look at neurons next to it, very close to this neuron, they will have the exact same lattice pattern in terms of the periodicity of the lattice and the orientation of this grid, but they will differ from one another in 2D phase shifts. Okay, so there'll be a phase of the lattice, which is going to be uniformly distributed across all the neurons recorded. And then, in fact, if you go further in the brain, so this is now the region of the brain where these cells are found. So in a recording over here, there is a group of neurons, as I mentioned, over here that have the same period, the same spacing. And then, but as you move further along this axis, it turns out that smoothly moving along this axis, you find a discrete jump in the periodicity of the neurons. So you find another cluster of neurons that now have a distinct period of firing, but again, they share the same orientation and period as one another, and then there's another jump, and so on. Okay, this is a top-down view of the neurons recorded in cortex, because cortex is a surface brain, it's on the surface of the brain, and so these are just the positions of neurons. The open circles are just positions of the cell bodies, and these then are color-coded by which spatial period these neurons exhibit. They're also color-coded by whether or not they're grid cells. So yellow is one period, blue is another period, and red is another period. And you can see that these are discrete patches of neurons that have different periods. Okay, so the period distribution is discrete. You can see this, by eye as well. Now, these periods, okay, as came up earlier, are actually intrinsically determined. And it turns out that the periods that they're distinct are all quite similar in scale. So strangely, right, so the smallest period is about 1/3 of a meter, and the biggest period is about 1.5 meters. It's like less than 1 order of magnitude in scale. Okay, so that's surprising. Okay, and so the first question, that I asked, anyone I think would ask is, why on earth? Like, why position is a local and non-periodic quantity, right? The animal's position, and it's encoding this using a code that's periodic and non-local, right? Because each neuron is firing in multiple different locations in a periodic way. So what is the why, right? And okay, so what's really interesting is if we take a temporal analogy, it's kind of like walking into a room, and instead of seeing a clock like this, right, that tells you something about the time in a 12-hour day, instead you see a wall full of clocks. Okay, one of the clocks has a period of 9 minutes, 10 minutes, 11 minutes, etc., like all very small periods. Okay, and why are these all small periods? Well, these are like the grid modules, but the periods are all small because 1/3 of a meter to 1.5 meters is very small compared to the linear distances rats run in a day. They will, in natural conditions, go foraging over 1 kilometer per linear dimension per day. Okay, so it's like this is the scale that they care about is kilometers, but these are the periods that they exhibit. Okay, and so we can define the code for space, kind of like thinking about this code for time, where if you think about x as a one-dimensional spatial variable, then instead of representing x directly, these grid cells are representing, a group of them with the same period lambda 1 is representing position as just a phase modulo this periodic lattice lambda 1, and the next group is representing position as another phase modulo this other period lambda n, and so on. And there's just a handful of such modules. So n is of order 5 to 10. They're just 5 to 10 discrete periods. And this is our code. Okay, and so, right. So it turns out, and I'm not gonna spend a lot of time because I'm interested in this question of emergence, but we'll come back to this coding question. But it turns out that this code is actually very interesting and quite beautiful. So let me just very quickly try to walk you through this. Okay, so now imagine each of these phase variables, right? Like, because each group of neurons that has a periodic response, their response completely repeats once the animal has moved by a distance lambda 1, right? And this group repeats at lambda n. So let's just consider a code with two modules, right? There's two groups of neurons with two periods. Lambda 1, which is the size of this axis on this torus, and lambda 2 goes around this way, okay? So you've got one phase that wraps around 0 to 1 here, and one phase that wraps around 0 to 1 here. And now let's assume that position x is varying along this axis over here. And so we just started some position on this phase coding torus, this biperiodic thing for the two modules. And as position increases, we start from here, and this coding line wraps around. Because the two periods are not the same, when it wraps around, one period has wrapped around, but the other one has not. And so this line does not close on itself. So it keeps going, and it keeps going. And so it turns out that for generic choices of period spaced within this interval from the smallest to the largest period, this code defines a space-filling curve. And in fact, again, for generic choices of the periods, you get this very nice interleaving space-filling curve where nearby positions, colored with similar color here, are represented far away in the coding space, right? So nearby physical positions are represented far away in the coding space, and nearby points in the coding space represent far positions. Like you can look at, the blue and the red are nearby, even though they're very far away in real space. Okay, so this coding, the system has this very interesting coding property where, by virtue of being a space-filling curve, you get unique code words. So the range of positions x for which you have a unique code scales exponentially in n, the number of distinct periods. And moreover, this interleaving property actually gives it a beautiful property of error correction, which I will not talk about in any detail, but I can refer you to papers. Okay, so let's come to the question of mechanism, and let's first talk about hypothetical mechanisms for a group of neurons that have a common period, or in other words, one grid module. Okay, and so the picture actually is quite simple. So let's assume that this is a sheet of neurons. So this is cortex. So this cortex has laminar organization. Imagine that this whole black square is a dense sheet of neurons. And let's just consider one neuron here in the middle, this white one. And let's consider ansatz that this neuron connects to its neighbors with this center-surround profile of interaction where it excites its immediate nearest neighbors, inhibits everybody in a ring around that. Okay, so if this connectivity is strong enough, and if all cells have the same connectivity to their own neighbors, right, in the sheet, then you can show that, okay, so what will happen? Well, so just conceptually what's happening is, if this neuron happens to be activated just by random chance, some fluctuation, you start with homogeneous mode activation, there's some fluctuations. If this neuron is more active than usual, it's going to more effectively inhibit all the neurons in a ring around it. But then the neurons just on the periphery of that inhibited region are no longer being inhibited because their inhibitory neighbors are silent, right? So some neuron on that periphery is going to be able to come on. Let's just say that one, by symmetry breaking, comes on. And now that neuron is inhibiting all the neurons in its ring around it. So the neuron at that intersection is maximally disinhibited. And so it's going to turn on, and so on. So you get pattern formation. And if that interaction was circular symmetric, then the pattern that you get is a nice equilateral triangular lattice. Okay, so now if we look at, so this is a sheet of neurons, as I said. If this were a 100 by 100 neuron sheet, it's 10 to the 4 neurons. And it turns out that the states, these states, right, so these pattern forming states are stable states. So with 10 to the 4 neurons, you get a stable state here. And in fact, there was nothing special about that particular phase of the pattern. All translations of that pattern are equivalent fixed points of the dynamics. Okay, so that defines a torus of fixed points, which is a 2D surface, right? Because the pattern, if it flows by one period in this dimension, you're back to where you were. And similarly, if you flow by one period in that dimension, you're back to where you were. So the set of all dynamical fixed points of the dynamics of this neural circuit lie on a two-dimensional manifold. That's a torus. Okay, so this is amazing. It means that you look at 10 to the 4 neurons, a collection of 10 to the 4 neurons, and you measure their states, and the prediction of this model is that they will always lie on a 2D manifold rather than being anywhere in this 10 to the 4 dimensional activity space, state space. Okay, that's actually breathtaking that such a thing could be possible. Yeah. That's right. That's correct, and that's a very good point. I'm glad you raised it. So actually, this is not yet explaining the phenomenon of grid cells. That's exactly right. This is a different space in which we're visualizing activity. This is actually the population of neurons and their instantaneous response of the whole population. And I haven't shown you any single neuron response as a function of the spatial variable, right? So then, in fact, we need, that's exactly where I'm coming, the next step. So ansatz number 2 is that as an animal moves in 2D physical space, it has an estimate of its velocity, and somehow there's a coupling from that velocity input, for which there's actually also a very simple mechanism, which I can't get into because of time, but you can, again, I can point you to the paper. But let's just suppose that this pattern, right, like all translations of this pattern are equivalent fixed points. So let's suppose that as the animal moves, say, east, this pattern flows in some corresponding direction in the neural sheet. Okay, say it flows that way, and if the animal moves north, the pattern flows that way. So now as the animal moves, then what happens is that, like, now if we're recording a single neuron, say this neuron is being recorded, and it's active, okay, at a certain position, now the animal moves, so the pattern moves. Animal moves, pattern moves, animal moves, pattern moves, and the neuron was off, right, because the bump, so the pattern moves, so now that neuron has gone off, and now it's off, 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 on. Okay, so that then converts into a spatial tuning of the neurons. So as the animal's moving, this happens. If the animal moves faster, the pattern moves faster, and therefore the spatial period is invariant to the animal's speed. Okay, so you get that, yeah.
Can you just quickly describe, you mentioned fixed points of what dynamics?
Oh, so these are, okay, I didn't write down the equation. So the canonical neural network equation for everything that I'll tell you. Let's see, eraser. Oh, I can just use this spot. Okay, so it's tau s dot plus s. These are vectors, right? This is the state vector. So let's say n by 1 dimensional for n neurons. Okay, instantaneous. So this is S of t is equal to f of some w s of t plus some b. Okay, so that's our canonical neural network equation, and what we just talked about here are the couplings, the weights, right? So that's the W's, that's the interaction between the neurons, and then S are the activity patterns, and then B is any inputs. Here we're just assuming that B is a constant to all the neurons. Okay, except for the velocity, which is actually the thing that's gonna allow that thing to shift. Okay, so you know, this is all old stories. And so it turns out that, you know, this very simple model has been quite successful, like phenomenally successful in making, so it makes a lot of predictions, right? This was something that, you know, you look at single neuron firing, and because it's so it's so regular, right, and has this property where the circuit has to estimate position to be able to generate these firing patterns. So you get we already know that there has to be pattern formation in this circuit, and we also know that the circuit has to be able to convert velocity signals into positional signals. And this network, by actually translating in response to velocity, is intrinsically doing two-dimensional path integration, or two-dimensional integration, right? Like vector calculus is what's going on in the circuit. And so you can make, because it's now a circuit model, you have all kinds of predictions about population dynamics in the network. And in fact, experimental studies have now directly observed this two-dimensional torus in the state space. Okay, but my agenda here is not to tell you the old stories. My agenda is actually to tell you about something newer, which is multiple modules and how they emerge together. Okay, so so this was already a form of sort of emergent dynamics. You know, you have like this simple translation invariant connectivity, and then you get these activity patterns, and now you get these kinds of patterns which are never imposed on the circuit from the external world. This is internally emergent dynamics that is then imposed from the brain onto the world, right? You're imposing a lattice, you're like laying a coordinate system onto the world as you move around in the world, which is pretty cool. Okay, so one note is to say that I told you about this very special connectivity profile, you know, center surround, center excitation surround inhibition, to give you these kinds of patterns. But in fact, there's an infinite set of patterns that will give you the same kind of triangular lattice, and you know, it satisfies very simple conditions, which one can look at. But basically, there's a huge number of patterns. As long as there's some competitive interaction locally, you will get these kinds of triangular lattices. Okay, so now the mystery. Okay, so now we have a model which actually has been, you know, around for a couple of decades almost, and, you know, extensively verified. So then the question is, you know, how does the brain actually form these multiple discrete modules of grid cells? And is there something that we can understand about the dynamics that lead to these modules, where they're placed, et cetera? So the surprise to us when we were trying to understand this question was that if you look at the biophysics of the neuron, so this is an excised piece of that entorhinal cortex, this is layer 2 and 3, and you can do intracellular recordings to find you know, whether there's a discrete jump in the biophysics of the neurons, you know, in the different modules that allows them to have different periods and different properties. And it looks like if you, there is actually a variation in the properties, the biophysics of neurons, as you move from the dorsal to the ventral end, where the periods are changing, right, in the stepwise way. But it looks like those properties are changing, as far as we can tell from the recordings available. They're changing in a graded way. Yeah. I'm sorry. Oh, PV terminals. Yeah. This is a measure of the width of the arbor size. So PV neuron arbor size terminals. It's their radius, and so this is something that's kind of smoothly varying as a function of the dorsal ventral position along this axis, and also other properties like the membrane time constant and the threshold of neurons is also varying seemingly continuously, even though the functional properties of these neurons is jumping in these discrete ways. Okay, and each of the networks, by the way, because their periods are incommensurate, they're different from one another, they can't be coupled in their dynamics, right? Each one is advancing at its own rate as a function of the movement of the animal, and so the dynamics must be uncoupled. Okay, yeah. That's right. So this is all the grid cell circuit, and this is actually, I put this on here because this is morphogens, exactly. And so, and in fact, the reason I put it on there, so thanks for the question, is to give you some other examples of, you know, starting from continuous gradients in a biological system and ending up with something that's modular or segmented, right? And in this case, there's a very a specific property where you've got sort of these morphogen gradients initially, but then the thing that specifies, right, this is like the question we started out, this philosophical question of how much information do genes carry. So in this Drosophila segmentation case, basically you have a downstream gene for each of these segments that is reading off the concentration of this gradient and then deciding to become active if the gradient hits a specific value. Okay, so in other words, this modular structure is really specified genetically because it's a gene for each segment, okay? And here the question is there a non-genetic way to get, to get this modularity to arise? Okay. Okay, so then you might say, all right, so the first thing, because this is a, this is a very simple pattern forming system that we've just understood in the context of single modules, the natural thing to say is, let's just take that model and let's just change something, one of the parameters of the model, according to this gradient, right? Let's just grade one of those parameters, right? And maybe the pattern forming dynamics, maybe the dynamics force the pattern to be coherent. Maybe they want it to be, it needs to be locally coherent, but of course the graded pattern makes, a pressure to actually change the pattern along the length. So maybe we'll see discontinuities or steps in the pattern just from that process of like wanting local coherence and local translation invariance, but with this more global parametric variation. All right, so let's do that. Oh, so what you see is, you see that you get actually a graded pattern that forms here. So this is the pattern. This is now just quantifying the spacings, and you can see that this is this is a 1D model, this is a 2D model, and you can see this is just quantifying the periods, and you can see the periods are just smoothly changing across this. Okay, so gradation in the lateral interaction only produces graded periods in the form pattern. So unfortunately, this is not the solution, and we can understand this very well. You can just do some very simple dynamical, some analytics of the dynamical system, assuming that this gradient is changing sort of adiabatically. So you can just assume that any given position, the value of the parameters is fixed. So then you can just solve the system like a standard Turing linear instability system with pattern formation locally. And in fact, we also predict analytically that you will get patterning with the graded period. Okay, yeah.
Just to be clear, on a previous slide, what would modular success look like in the sense of what would you expect to see?
So modular success would be that, it would be just like the experimental data I showed you, whereas as a function of position here, you would get a flat period, and then you'd see a jump, and then you'd get another flat period, and then a jump. Okay, so that would be modularity, right? Group of cells with identical periods, and then a jump, a group of cells with identical period and a jump. Yes. Oh, yeah, that's right. So it's modularity on two levels. So one modularity is going to be the pattern formation process, right? Which I guess you can think of as it's dynamical because it's some dynamical fixed point, right, of the system. So yes, it's dynamic on that level, and then it's also going to be modularity in terms of function. I'll show you. So. Not on the level of the physical connectivity. That's correct. Yeah. That's right, but they've got a separate. Each module has its own devoted genetic cascade, right? Each module has a readout. Like there's no non-genetic process that can give rise to this, as far as I understand, right? So you're saying that there are existing models that start from a continuous gradient and then do not have discrete different genetic cascades. Okay, they do, but then I would call that a genetic mechanism, not a self-emergent mechanism from. Yeah, but it's genes, right? Like the gene has evolution has made this gene be there that has set a fixed threshold and all of that stuff, right? There's I mean, maybe it's semantic, I think. Like to me that would be like genetically coded, But, and what I'm looking for is, I'm looking for something where there's no genetic involvement other than in setting up the gradient and maybe in some other simple rules. But there's nothing that's instructing where modules should form. There's no genetic instruction on where these modules should form. So I think that's, yeah. But of course, I mean, even, the process of genetic thresholding, et cetera, I mean, and the cascades are going to be dynamic. So I totally agree with you. Okay, so that's all you get. And so then the question might be, well, okay, maybe, okay, so we're looking for two scales of structure, right? So there's like the local patterning, and then there's the global modularity. And so maybe we need scales, right? Maybe this is only one scale of interaction and it's graded. Maybe one needs to throw in a second scale, right? But what does that mean? Because I already showed you earlier that there's actually an infinite set of these coupling matrices that can all give rise to this local patterning, and each of those kernels can have multiple scales in themselves. So, right, the process, in other words, this process of local patterning is not sensitive to all the different, scales that are present. I could throw in a second scale in this kernel, and it's still just going to lead to the same thing. So what two scales mean, the only way that you can differentiate scales is to say one thing is graded, and maybe there's a qualitatively different thing, which would be another interaction that's not graded. Okay, so I'm gonna just add in a second interaction. Okay, so now let's assume we've got two interactions, and I've just changed the shape just for fun. Like, it doesn't matter. It could have been, both of these could have been that difference of Gaussian shape. But one of these, the green one, is graded in scale. And the other one, the orange one, we're gonna assume is fixed in scale as you move here. Okay, so now these two interactions are changing relative to each other, right? Because one is changing in a graded way and the other one is not. Okay, so and the shapes don't matter. So the moment I put that in, okay, let's just see empirically first what happens. And you can see that instantly the pattern formation process gives us a bigger scale here and progressively shorter periods. We can then look at, I can just quantify the periods, and you can see that these discrete steps. And in fact, there's also functional modularity. So using the mechanism of velocity input into these networks, into these modules, I give the same velocity input to all of the neurons here. And actually, what you can see, and the lighting isn't great, but basically this is a 1D version, of this network. And now on this axis, I'm plotting. So each stripe here is this network state at one moment in time, and the different rows are different moments in time. So what's going on when I give a velocity input is that these stripes, these bumps are moving. So then we get these diagonal stripes, right? That's moving bumps. And what you can see is that these different bumps are moving at different rates. You really can't see it. Is there a way to turn that light off? How do I do it? Yeah. Okay, but basically, yeah, I just want to show you this because, maybe, does that help? Maybe not? Maybe not too much. So you can see these diagonal stripes here, right? It turns out there's diagonal stripes here that are narrower, and these are even narrower, and so on. Okay, so, and you can see that there's sort of this interface over here where there's a step, and you can see that there's actually a slip, right? So these bumps are moving. That's what these diagonal stripes are. These bumps are moving, and they're moving at different speeds. So they're actually slipping past one another, right? These bumps are actually sometimes coherent and sometimes not. So they're actually slipping. So these networks now, even though they're physically one network, these modules are kind of integrating the velocity completely independently of one another. So they've become not just independent in terms of having these different pattern periods, but they also have independent velocity responses. This is a 2D network. It's the same thing. Okay, so what's happening? Can we understand this, right? I just told it to you like it's magic. So if we look at the early time scale dynamics here, okay, so you can have a quick look. This is really early time scales. We start from the homogeneous mode, and you can see the pattern forming, right, locally. You can see the periodic pattern. And I think what's really interesting and which allowed observation that allowed us to solve this problem is that basically the different periods are forming in situ, right? So in other words, the modules are forming at the same, you can already see that the periodicity locally here is distinct from the periodicity here right from the outset. right? So the local pattern formation, it's not like you first get a local pattern formation that's similar period everywhere, and then you refine the module periods as being different. It's just, it starts right from the outset as like different periods and local patterning. Okay, which means that the process is the same local instability process that gives rise to local patterning also is the thing that gives rise to the modularity. So we can then try to understand this process. So we can just do the same analysis, just linear perturbation analysis in Fourier space to find like the unstable modes that give rise to these patterns. So yeah, so it turns out that it's pretty understandable. How much time do I have? Oh, only 5 more minutes. Oh my God. Okay, 10. Okay, yeah, I'm kind of behind. Sorry. Okay, so let's try to understand this process. So what happens? I should just backtrack just a bit. So here, okay, so these are the neural network equations that I wrote down here on the chalkboard. Okay, and what we do here to find the pattern solution, right, is we first take this nonlinear system and we linearize it. Okay, so we linearize it around uniform activity state over here. And so this is just a Taylor expansion of this on the right side. And now we just look at the Fourier transform, right, of this. So the Fourier modes, because this has translation invariant connectivity, so the natural thing is to look at the dynamics in Fourier space. And so we just take this linearized equation and transform it into K space. And so in this Fourier space, right, so these, another thing we've done is we've gone from like discrete space, right, these weight matrix that multiplies the activity vector, and instead just in the continuum, the matrix multiplication becomes a convolution function, okay? And the index I and j for the neurons just becomes a spatial coordinate x and x prime, okay? So first, so we're doing 2 steps here. We're taking the continuum limit, and we're also linearizing this nonlinear dynamics around the homogeneous fixed point. And then we're looking in Fourier space. And then the solution to this problem, so the modes, so this is now an equation for the dynamics of the Fourier modes, right? And the Fourier modes that are going to grow fastest are the ones with the biggest eigenvalue. Okay, so in other words, this network is going to have a pattern with given whose period is given by the inverse of k star, where k star is the argmax of this Fourier transform of the coupling matrix. Okay, and so that's the way one finds the solutions. And so we can then just, so everything depends on the Fourier transform of the coupling matrix, right? So now let's just consider that we have these 2 weights, right? One narrow one, and then this broader but fixed one, right? And or we could have it be this shape, whatever, it doesn't matter. So the wider interaction term is the orange one, the narrow one is this green one, right? And the green one is graded, so as you move from dorsal to ventral, you get this change in this green interaction, but not in the orange one. Okay, now when we go into Fourier space, the wider potential has more wiggles in the Fourier space, right? And the narrow interaction term has broader structure in the Fourier space, right? And so basically the broad one, which is the static interaction, has like this narrower profile in the Fourier space, and it's periodic over here. And as you move dorsoventrally, then the smoother term in the Fourier space is now moving, okay, as you move dorsoventrally. So basically what happens now, if we consider the combined, and we can replace, so basically this whole dynamics of pattern formation can be thought of as descent on this energy landscape, where the energy function is just the sum of these 2 Fourier components of these 2 interaction terms. So if we look at the solution is basically the max of the sum of both of these W's, okay? And so you can see that what's happening now in this small animation is as you move from dorsal to ventral, right, in this space, then if we just look at the position of the maximum, well, the maximum is always at one of the peaks of this oscillatory term, which corresponds to the static wider potential. But then which of these narrower peaks is selected as the global maximum depends on the smoothly varying interaction term that is actually changing, right, from dorsal to ventral. So we call this a peak selection process where there are these predefined peaks, and then which of these peaks is the solution is set by by this narrower interaction that's graded. Yeah. So the right. So the you mean here? No, so this is just simple pattern formation, right? So what you've got here is we've just got this is just an equation that's just telling you, right? So this is just a dynamical equation, linear dynamical equation. We can decompose it into Fourier modes, right? And what this is just saying is if you look at the Fourier modes, right, of the W matrix, the states, these epsilon, the perturbations that are going to grow fastest are the ones that have the biggest eigenvalue, right, in the Fourier space. So it's your classical pattern formation theory. S here is neural activity, and then this is the linearization of the neural activity around some homogeneous state, right? And so that's why you just get, it's just like growth. It's just linear instability, right? Which is selecting like the K, the Fourier mode with the biggest argmax in the W. Yes. So that's exactly where I'm going from that. Okay, so this is it. Okay, so this is conceptually how you're getting. So like this is a mechanism for modularity emergence, which is peak selection, which is coming from one fixed interaction that is slightly larger scale and one graded interaction that is a slightly narrower scale but graded. Okay, but they're both completely local. And so let me now quickly run through the results. And so thanks, Gautam, for the question, and I will pay you that $5 after the talk for raising it. But it turns out that, okay, so what's going on is the peaks. You see the peaks, most potentials that are, have some sharp cutoff and are, wide will have a dominant Fourier mode, which is, just going to be like close to a pure sine wave. Okay, and so the peaks are going to be like integerally spaced, right? It's just going to be multiples of the period. And so it turns out that this model predicts that your successive solution, so the periodicity of the pattern in the mth mode to the m+1th mode, or module, okay, is going to be basically integer ratios, m+1 over m, with a small, correction. Okay, and that small correction is the only thing that depends on the detailed shape of the interaction, and otherwise none of it depends on the shape of the interaction. So this model is predicting that your successive module period ratios look like something like m+1 over m with a small correction. Okay, and so we can take the mean value of those ratios for small m's, right? So m equals 1 through 4, few modules, and we get a number that's like 1.37, and it turns out that the measured value is 1.4. But we can do more because, here we have, this model is saying that actually the period ratio shouldn't just be some average value. The period ratio should in detail change as a function of m. So the second to the first module ratio versus the third to the second module ratio should be different, right? In this specific way. Okay, so what do we get? So in fact, so I should just, wait, what happened? Oh, maybe I put that later. Oh, yeah. Okay, so I'll come to that. Okay, so we'll come to the, but anyway, so that's the prediction, right? So that's a strong prediction. So we'll come to that. Okay, how many modules emerge? Oh, I want to tell you, yeah, okay, so it turns out we can analytically compute how many modules emerge, and it turns out that the number of modules that emerge does not depend on either on any of the widths of the lateral interactions themselves. Okay, they don't depend on the size of the network, right? Here's the expression. So D is the width of the broader interaction, sigma min is the narrowest of the graded interaction, and sigma max is the widest part of the graded interaction. And so it's really like the ratio of the fixed scale and the varying scale interactions. And it's the difference between D over sigma min minus D over sigma max that tells you how many modules form. So it's really some property that is not dependent on these microscopic parameters, right? It really just depends in this, very simple way on these combined properties. And so if you keep the maximum width and the minimum width of the graded interaction fixed, but you change the shape of the gradients, it does not affect how many modules, because if sigma min and sigma max are fixed, but you change the shape, you still get the same number of modules. The only thing that changes is where the module boundaries are relative to one another. Okay, and the other prediction is that, you know, as long as you, so that's the only thing that changes. And so there's kind of this like topological robustness, right, in all this process, because the shapes of the connectivity do not matter. Okay, the individual scales of things don't matter. The size of the system doesn't matter. Okay, and so it's just almost independent of everything. So what sets the module size? So it turns out that because the number of modules is independent of the size of the system, right? So if you double the size of the system here, or triple it, you still get the same number of modules as long as you hold the sigma min and sigma max fixed. Okay, so that means that if that's true, then it means that the module sizes are themselves doubling or tripling, right? So this is a system then that has self-scaling with system size. So there's like, you don't need to change. So the local interactions, which are the only thing giving rise to both phenomena, local patterning and global modularity, and they're all local interactions, okay? They do not set, their scale does not set the scale of the size. The module size is extensive, which is, you know, kind of surprising. Okay, so in other words, then what this is saying is that, you know, even as, you know, you change, you know, these grid cells are observed across various species, it's saying that you can still get the same number of modules across species with brains of very different sizes without having any, without having any need for genetic variation, right? So you don't need any parametric gene expression pattern or anything that should scale with the brain size. Okay, so basically you get a whole host of predictions from this model. You can have developmental predictions, transcriptomic or connectomic or physiology predictions. So the idea is that you can get predictions about obtaining spatial patterning in the cortical sheet, that can arise without any change, right? So in this model, one thing that's interesting is that you can imagine that if there's a chemical that sort of sets, so sorry, there's one more thing I should tell you, which is that this parameter that I varied was the width of the interaction, but you could also instead vary the time constant of cells along this axis. Okay, and then you get identical modularization. Okay, so that means that you could imagine that there's some kind of neuromodulator that over development, you know, kind of ramps up, right? And if it ramps up, you can go from having a single module early in development, and it could suddenly give rise to 2 modules and 3 modules, and so on. In fact, the really interesting thing is that an experiment from the Moser lab, the discoverers of the grid cells, that isn't published yet, found that on day 9 post in day 9 post birth of mice, there's 1 grid module, and then on day 10 there's suddenly 2, and on day 11 there's 3. Right, so it means that there's no time for rewiring, right? So it kind of suggests that there might be a very rapid process that gives rise to emergent modularity rather than having gene cascades and things like that. So that seems suggestive, and that's interesting. Okay, I know I'm close to, I'm past time here. So okay, there's also, you know, there's a whole bunch of, you know, predictions one could make, you know, depending on your interests. But let's just talk about the period ratio prediction that Gautam asked about. So as I said, you know, one predicts specific sequence of period ratios in this model, and you know, so the first pair would be 1.5, right, 3/2, and then it would be, you know, like, so you'd have 3/2, 4/3, 5/4. So this is the sequence of predicted period ratios, assuming that 5 factor was 0, and then here's the known parameters in individual mice, 1.5, 1.3, and 1.2. So it seems, you know, suspiciously close. It's kind of crazy that this is the case. So this was encouraging to us. All right, okay, so I will just basically just, you know, sort of conclude by coming back to the sort of philosophical view where, you know, we talked about Drosophila patterning and segmentation, where you've got a gradient and then you have different downstream gene cascades that are reading out or thresholding that value of gradient. So you kind of have these gene cassettes that lead to physical cassettes, right? And then we've got kind of pattern forming dynamics where local interactions can give rise to patterns. But the problem with Turing pattern formation models is that in Turing pattern formation models, the scale of the pattern is completely determined by the size of the interaction. So whatever is the interaction scale is your pattern scale, and you get the same pattern scale everywhere, okay? Now in this mechanism that I've told you about, this peak selection mechanism, it's possible to get the coexistence of patterns of multiple scales, and it's also possible to get structure that emerges from purely local interactions. You can get really global structure. Okay, and so that's and then a question for Drosophila or other developmental biologists is could this kind of process also be acting in these gene cascade-based positional information models to robustify the spatial information process? And I'll just mention but not describe that this same mechanism of peak selection, where you've got sort of this rough, you've got two landscapes, a rough landscape which is fixed, and then a smooth landscape which moves as a function of some parameter. So as theta varies some parameter, the position of the smooth landscape changes. Then you're automatically using the smooth landscape to select one of these peaks in the rough landscape, and you will automatically get the step-like modularity in the solution of any dynamical system. So we can apply this to ecological models and get basically from smooth gradients like sunshine and moisture, you can get in an interacting web of species, you can get discrete niches. So you go from like uniformly or graded expression of populations of species to getting these like self-organized modular niches. Okay, so thanks so much, and I can be available for questions later. Go ahead.
So if I understood correctly, the reason that you get this 3D manifold is because you start off with the neurons already embedded in 2D.
No, because, right, I mean, that's not necessary at all, because you could have neurons embedded in 2D space, but then their interactions could be like a Hopfield network, right? They could have been random all-to-all interactions, then you would have much higher dimensional activity. You could also have, anisotropic or otherwise inhomogeneous connectivity between neurons. So the reason you got 2D dynamics is because you got 2D pattern formation, which came about because of the symmetry, translation invariant symmetry in the connectivity. So, so we set up connectivity that had the symmetry of 2D translation invariance. And so is that. So it's not simply organizing neurons physically in 2D space, but it was actually setting up the connectivity to have the symmetry in 2D space. So, I mean, I think because the cortical sheet is a sheet that makes it very easy for the brain to set up this 2D translation invariant connectivity. But other mechanisms can also contribute, even if the brain were 3D, it could be if this was really ethologically necessary, it could have come up with that. In fact, the part that I didn't have a chance to speak about was training, random all-to-all like recurrent neural networks with no spatial embedding to do some very simple ethological tasks. Like basically the task is you give the network velocity inputs and you ask the network to just generate a self-consistent notion of displacements, which means if the animal stays at the same position, the network should return the same code, whatever code it chooses to use, but the code shouldn't change. And if if you give it velocity inputs corresponding to a displacement, the code should change, right? So basically just saying if there's no change in position, there should be no change in the code. If there's a big enough change in position, there should be a change in the code. And if the velocity trajectory is a closed loop, so the animal returns to the same position after some excursion, right, or the agent, then it should come back to the same code. That's it. Those three, okay, so simple conditions of like self-consistent, internally self-consistent coding. If you do that over large enough spaces, gives grid cells as the solution, so we have no 2D embedding in any of it, right? So there's a learning mechanism that could also give rise to it. It's just that by having everything be in 2D, maybe biology is relieved of having to learn all of that, right? It can just, I mean, you just have simple growth rules for each neuron that just, form, each neuron, all it has to do is literally have a spatially confined arbor that's statistically isotropic, and you're gonna, you're gonna be most of the way there. Yeah.
Spatial structure, do you actually see
Oh, yes. Yeah. So in fact, yes, that's a wonderful question. In fact, I went over it quickly. Here it is. So great question. So this is an experiment from the group of David Tank. I already described this. This is a top-down view on the tangential, this is the tangential surface of cortex. And I'd already showed you the coloring of these different modules, right? So this is the different modules abut one another, which is also, predicted by this, gradient-based model, because the angle of the module boundary should be close to orthogonal, which it seems to be. Okay, but then if you look within a single module, so this is within a single module, now we're coloring cells based on their phase, right? So all the cells have the same periodicity. We're coloring cells to be the same color if they have similar activity phase. Okay, so all the greens have the same activity phase, and you can see that cells that have the same phase are next to each other, but they also form some discrete clumps. And the same thing is true for the orange ones, right? There's like one orange clump here and one orange clump here and one orange clump here. And so I think if you squint, you can see that the sort of green clumps spaced apart and the orange clumps are spaced apart by about the same amount. So we compute the relative physical location in Y and the relative physical location in X of cells that have similar phases. You get this plot over here, and we compute the autocorrelation of that. You can see it's close to a triangular lattice. So basically, the cells seem to be on a physical triangle. You can see the activity pattern as you would predict from the spatial patterning idea. So that seems to be true.
I remember seeing that from Modular, but they showed that the ring attractor that wasn't really embedded.
Oh, in the ring, well, so the ring attractor is in the state space dynamics. This is like literally on the cortical surface. There's also a. In the fly, it's a physical ring, and what we're showing here is that it's actually a physical grid.
Yeah, so what I'm saying
in the grid cell system. It need not have been, because for example, the mammalian head direction circuit, which is a ring, the state space is a ring, but the physical placement of the neurons is not a ring, as far as we know. So yeah. Okay, should stop.